So today we'll be talking about the cycle of the HIV virus. So we were done with the structure of the virus. Now let's talk about the structure of the virus. Basically, the cycle of HIV consists of many steps. Many steps to multiply in our body. There are many steps. Number one is attachment. Second is fusion. And third is the reverse transcription, and the fourth one is the integration, and the fifth one is the replication. We'll be talking each steps in detail. And the sixth step is assembly, and the seventh step is budding. Okay, budding will be the last step, and first one step is the attachment. So at the first one step, the virus attachment. So at the first step, the virus will attach with a, as you guys all know that they attach to the T helper cell or T lymphocyte cell, because of they are having a receptor that is CD4, so that can easily identify their, you can say, their host cell. So the host cell is the T lymphocyte. So virus attach with a T lymphocyte cell. Okay, so with their cell they are having, as you guys all know that the HIV virus is having the glycoprotein. So their glycoprotein, they just enter into the host cell T lymphocyte with the help of glycoprotein. Glycoprotein is the protein that is on the HIV, on the surface of the HIV virus. So they are having the glycoprotein for the attachment. So the glycoprotein helps them to attach with the lymphocyte, which is the host cell of the, you can say that the host cell of HIV virus. So they help the glycoprotein helped to attach with the lymphocyte. So the first one is the attachment. So the virus is now attached with the lymphocyte. So second step is the fusion. So while the remaining part that are attached with the glycoprotein will be on the surface of the virus, just the remaining part will be injected to the virus, that is capsid. So the capsid will be attached, or you can say that will be enter into the host cell. Their spikes will be remain out. They are just capsid, which is the main capsid that is having the RNA. HIV is having the single stranded RNA, the double stranded RNA. So they will be moving inside the host cell and enter into the host cell. Okay, how fusion occurs? Because there in glycoprotein there is a protein. So they are having you can say that they are like having a protein. Okay, so the glycoprotein consists of GB41 and GB120. So what are these? They both are having they both are the glycoprotein spikes. So GB41 is for the fusion. So GB41 will be doing the fusion to enter into the host cell. So GB41 will be helped to the HIV virus to inject inside the host cell. So that will be, we are done with the structure. So that is the attachment, or you can say that there is the attachment, that is the fusion. That virus will be inside the host cell. Only the capsid of the virus will be inject on the glycoprotein will be on the surface of the viral protein. They will be not injecting. So now the third part is the reverse transcriptase. So what is reverse transcriptase? Reverse transcription. Reverse transcription is the step where the viral RNA, that is the capsid of the HIV will be making a new DNA. This is basically an enzyme that is present inside the HIV virus, into the you can say inside the capsid. So they are having the enzyme that will help the RNA, and RNA, you can say that the viral RNA that is inside the HIV, the RNA will be converted into the DNA. So that is called the reverse transcription enzyme. That is the step where the HIV is having the viral RNA. So uh, they will be you can say that this is basically the viral RNA will be making a new DNA. This process is called the reverse transcription. Okay, so the the you can say that the enzyme that is used for this is the reverse transcriptase. Reverse transcriptase is the enzyme that through which the RNA will be replicate into the you can say that RNA will be changed into the DNA. So that is the enzyme which is reverse transcriptase, and the reverse of the RNA into the DNA, the changing of the RNA into the DNA is called the reverse transcription process. Now the fourth step is the integration. Now what is integration? Okay, so integration means that the DNA which is like now the HIV is having the DNA. You can say that they are having a DNA. So they are having the capsid, they are having the genome which is DNA. Now because the RNA is changed into the DNA, so now the DNA will be entering into the host cell nucleus or host cell genome. So they'll be entering into the host cell genome, entering into the host cell genome. Okay, where they'll be entering the host cell genome. The host cell genome is the DNA, and the HIV virus is converted into the DNA, so they'll be also having a DNA. Now the HIV DNA will be, you can say that they will be like changed into the, you can say that they will be attached with the viral DNA. They'll be attached with the viral DNA that is having a enzyme that is called the reverse, you can say that the with the help of integrase enzyme. So the HIV is having the reverse transcriptase enzyme, and you can say that integrase enzyme. With the help of integrase, the DNA of the HIV will be attaching with the DNA of viral genome, which is the host cell. So the viral DNA, that is the HIV genome will be attached with the our body host cell that is having a genome also DNA. So they'll be attaching with that with the help of integrase enzyme. So here the process is that they will be attaching with the help of integrase enzyme which they are having, HIV virus is having. So they will be with the help of integrase enzyme, and they will be attached with the enzyme that is called the condition that is provirus. They will be inactive for the several years. Okay, so now the fifth step is the replication. Okay, so the replication is that there is a process that is replication, and the replication is that the DNA will be making the mRNA with the help of the DNA. Okay, now the integrated host DNA will be making a mRNA. Now the host DNA through which the HIV is attached with that, so their DNA will be making the mRNA. Through mRNA they will be making a protein. Virus DNA, that is the HIV DNA, will be making a protein with the help of the viral DNA. So that through the DNA the mRNA will be produced, and through mRNA there will be the protein will be produced. with the help of the using the host cell, that is the enzyme, that is RNA polymerase. The protein will be made by the RNA polymerase, that is also the enzyme of the HIV. So the HIV will having the three enzyme: integrase, reverse transcriptase, and the RNA polymerase. RNA polymerase will be they are having the enzyme to make the protein. So through the viral DNA they'll be making the through the viral DNA they'll be making their own protein. Now the sixth step is the assembling. Okay, what is assembling? When a protein is found by the HIV virus, or you can say by the viral through the HIV virus, because they are having a viral DNA as well, so the host DNA as well, so they have make the protein. So now in assembling protein they will be used the other enzyme, that is protease. They are having her protease enzyme as well. So they are having a protein they are having the enzyme, that is protease. Protease is a, you can say that they will be cut down the protein that is made by the replication. So they will be the small protein units will be the large protein units will be converted into the small protein units, or you can say the fragments. So the assembly is the step when the protein are formed by the HIV. They will be using another enzyme, that is the protease, to cut down the protein into the small fragments. So later on they will be joining with the HIV genome to develop the new virus. Okay. So assembly will having a protease enzyme. In assembly, you can say that the step there is a enzyme, that is the protease. Okay. So now they are, like, you can say that now they are having a last step, that is the budding. Budding is that the new progeny of viral will be get matured and they'll be connected with the cell. Okay. So they'll be connected with the cell, they'll be connected to the cell phase. And after the virus is completed with the protein, they'll be formed. Okay, so we were done with the 6 steps and the 7th step for remaining. Let me repeat that this was the attachment process in which the HIV was attached with the T lymphocyte, that is host cell, and in fusion the glycoprotein were outside of the host cell, and just the capsid was inserted into the host cell through the glycoprotein, that is GP41, that is for the fusion. Okay, so the reverse transcription, that was the process through which the viral RNA of the host cell was converted into the DNA with the help of reverse transcriptase enzyme that was present in the HIV virus. So through this virus they just replicate, they just transfer from this to this, so that is called the reverse transcription process. And the fourth one step was that the host cell was the HIV virus was connected with the host cell genome with the help of the integrase enzyme, that was also the enzyme of HIV, through which the HIV DNA was connected with the you can say that the host DNA. So with the help of integrase enzyme, and that was called the provirus. And now the replication process, in which I said you that through the viral DNA which is attached with the HIV virus, so that will be making so the HIV virus will be making the protein through the help of their DNA, and with the help of the HIV. No, the host cell is having the RNA polymerase. They'll be using their enzyme. The HIV will be using the host cell enzyme RNA polymerase, and they'll be making through the protein. And the sixth process was that the protease enzyme, that was the enzyme of the HIV, that was the protease, the protein that is made, they'll be breaking down into the small fragments of the protein. Okay. So there were the three enzyme: reverse transcriptase and integrase and the protease. Okay, so the seventh step is that when the then the complete virus is formed, and they'll be connected to the cell membrane after the fragments of the protein, and then they will be released through the budding. Then they will be released through the budding process. Through budding process. What is budding? Budding means that they will be released out of the cell of the T lymphocyte, that is the host cell. They will be released from the T lymphocyte, and then they will move to the another cell to infect them. So the new progeny was virus that will be mature into the cell, and they will be consist the cell membrane, and they will be make a form a small projection, and they infect the cell, and they will be like, you can say that the glycoprotein will be the part of the cell membrane as well, so they'll be having their own covering, so they will be released out with the cell membrane, and they will be infected another cell. So that was the life cycle, and that is part 2. So we are done with the cycle of HIV virus. What's the full form of HIV virus? That is the human immunodeficiency virus. Okay, so they just destroy our immune system if the body is having the HIV virus. So it's very dangerous for the immune system, because HIV directly attack on the immune system, because they are immune system is having the host cell that uh, they just, you can say, identify the host cell because of the receptors of the because of the receptor of the host cell that is present in the, you can say that, immune system. So they are having the receptor, that type of receptor, they easily identify the